known as the single-cell culture. It uses the pieces of established undifferentiated or fibrous callus that are transferred to the liquid medium in a flask or other suitable vials. The medium is continuously agitated at around 30 to 100 rpm by fixing on a gyratory orbital shaker. It is a significant method for the mass production of cells. The mechanisms of suspension culture are: the agitation of the medium exerts a mild pressure on the tissue, breaking it into smaller cells aggregates and single cells. It maintains uniform distribution of cells and cell clumps in the medium. It helps gaseous exchange between the culture medium and the air inside the culture vial, which we refer as the aeration of the cells. Alright, what are the starting materials for suspension culture? We can use fibrous cells with the cell density 5 × 10⁴ cells per milliliter or higher. We need the actively growing and typically synchronized with the respect to cell division cycle. The liquid medium, whereby the agar is omitted. We need to add different plant growth regulators such as oosin or cytokinin, nutrients, and precursor ratio. The advantages of suspension culture are: it gives much higher rate of cell division that may be exhibited by the cells in suspension cultures than in callus culture on agar medium, which means that the cell division rate in liquid culture is much higher than that of agar medium. The cells in suspension culture can form embryos, which are known as the somatic embryos, through somatic embryogenesis, which is then multiplied or induced into the plantlets via morphogenesis. On the other hand, the disadvantages are: the ideal suspensions should consist of single cell, which is very difficult to achieve. Therefore, most of the time it consists of small cell aggregates, which is the clumps of the cells. The cell doubling time in suspension varies with the system and also the period. There are a few types of cell suspension culture. First, single cell culture, batch culture, continuous culture, which can be classified into closed and open, synchronous culture. First, the single cell culture. It is mechanical isolation, which tailing the leaf to expose the mesophyll cells, followed by scraping the cells with a fine scalpel. Followed by the enzymatic isolation, in which the degradation of the middle lamella is occurred to release the cells using macrosome incorporated with osmoprotectant, which is the mannitol. There are few protocols for single cell culture, which are filter paper roughness technique, microchamber technique, Bergman cell plating technique, and microdrop method. Alright, for the batch culture, the cultures are continuously propagated by routinely taking a small aliquot of the suspension and transferring it to a fresh medium, which is about 5 times dilution. You will exhibit the sigmoid pattern of growth with 5 phases, which are lag phase, short exponential phase, linear growth phase, deceleration phase, and stationary phase. This is the sigmoid callus growth model curve, whereby for the first lag phase, the cells are prepared to divide, followed by short exponential phase, in which the rate of division is the highest, linear growth phase, whereby the cell division starts to slow down and cells expansion increases. The fourth phase will be deceleration phase, whereby both cell division and expansion are declined. Lastly, will be ended up with the stationary phase, in which the number and the size of the cells remain constant. This is the detailed phases of the suspension culture. For batch culture, the callus can be maintained continuously in exponential phase by frequent subculture, which is about every 2 to 3 days. Addition of conditioned medium, the used medium which the callus culture had been grown before, reduces the lag phase dramatically. Therefore, it is not a suitable system to study the cell growth and the metabolism due to the constant change in the pattern of the cell growth and metabolism, and also the composition of the nutrient medium, as well as the consequent absence of steady state of the growth are inherent drawbacks of the batch culture. Therefore, we have the continuous culture, which is the better approach as compared to the batch culture. It is periodic draining out the used medium and addition of fresh medium. For example, we take out the callus or we filtrate the callus. We put the callus into the new medium. So for the new culture, there is no used medium inside of it. This is to ensure the maintenance of the suspension are under steady state of the growth for long periods. The cultures are known as a continuous suspension culture. It is suitable for mass-scale cell culture with bioreactor. These continuous cultures are widely used in the research and commercial. There are two types of these continuous culture, which are the closed and open continuous cultures. For the closed continuous cultures, they have the spatial equipment whereby the addition of fresh medium is balanced by outflow of the old medium. The cells from the outflowing medium are mechanically separated and added back into the culture. The cell biomass continues to increase with age of the culture. For the open continuous culture, the addition of fresh medium is balanced by the harvest of an equal volume of medium, which including the cells that maintaining indefinitely a constant submaximal growth rate, which can be further split into two types. First, chemostat. The steady state of the cell growth is maintained by the constant inflow of fresh medium in which the concentration of chosen nutrients is adjusted to adjust the growth rate of the cells. To study the effect of nutrients on metabolism and cell growth. Another one will be turbidostat, whereby the inflow of fresh medium is controlled by an increase in the turbidity of the cultures due to the cell growth. A desired biomass density is maintained by flushing out the cells. The last one is synchronous culture. It is done by the starvation or inhibition. The cells are starved of the nutrient, such as the phosphate or nitrogen, or hormonal factors that are required for cell division in G1 or G2 phase. The cells are then transferred to the medium containing the essential elements which will then enter the division synchronously. The synchronization can be achieved by using the inhibitors of DNA synthesis such as 5-amino-uracil, FUDR, hydroxyurea, and thymidine. Alright, how do we determine the growth in the suspension culture? First, we can use the cell counting, treating with the chromic acid 5 to 8% or pectinase at 0.25%. The second method is use the packed cell volume PCV, whereby a non-volume of uniformly dispersed suspension is transferred to 15 ml graduated centrifuge tube and spun at 200 times gram for 5 minutes. This PCV is expressed as ml pellet per ml culture. The method is very common, which is the cell fresh weight. It is commonly done by wetting the callus after washing the cells with distilled water to remove the medium, draining under the vacuum or dry by using the filter paper to obtain the net fresh weight of the callus. Following, we can use the dry wet callus, whereby the callus is dry for 12 hours at 60 degrees Celsius, for example, until the constant weight is obtained. The cell weight is expressed as the per culture or per 10 power to 6 cells. The last method is the non-invasive method, in which we tilt the conical flask at 30 or 60 degrees, but you make sure to sanitize the angle every time when you take the reading for 5 minutes. After that, you measure the height of the sediment with a ruler. The change in the height of the sediment with the edge would represent the change in the fresh weight of the cells. Alright, after we have done the suspension for a long period, how do we know whether the cells are still alive? Therefore, there are some viability tests for culture cells. First, we can use the phase contrast microscopy, whereby the cytoplasmic streaming and presence of healthy nucleus will be observed. Or we can use the tedrazolium test, in which the respiratory efficiency of cells is measured by the reduction of 2,3,5-triphenyl tetrazolium chloride (TTC) to the red dye formazole measured through the spectrophotometer. Or we can use the fluorescein diecetate FDA test. The living cells will give the fluorescent green under UV. The last test is we can use the Evans blue staining, which is used as the complementary to FDA. The damaged cells will take up the stain, but the viable cell will exclude.